Hello, everyone, and welcome back to another one of our Meet a Scientist Tuesday lessons. Today, we have a very special guest, Dr. Will Richardson. Hi, Will. How's it going?
Good. Hi, everybody.
Yeah. So, Will is joining us today. Is going to give us a really special talk, a lot on some of the focused around the Tahoe area. Where many of us are based. Will has a Ph.D. from the University of Nevada, Reno, from the EECB. It's evolutionary, what's the second B?
Ecology, evolution, and conservation biology.
Exactly.
Or maybe the E's are, I forget the order, but those are the three.
And that UNR PhD program has a really strong program. We actually hire a bunch of our scientist mentors out of there. And so we're super excited to have Will join us from that. Will is also the co-founder of the Tahoe Institute for Natural Sciences, which is, if you don't know about it, a really awesome outdoor and science education nonprofit that works in the Tahoe region. And I think, I'm not sure how broadly else there, but Will's gonna tell us a little bit about that, which is really awesome, and some of his research.
Cool. All right, thanks so much, Spencer.
I'm going to turn things over to Will and hop out of the way.
All right. Okay. Should I share my screen now?
Yeah, go for it.
Well, actually,
yeah,
you need to toggle to that, and then I do this, and then we're good.
Exactly. You're all set. And
all right, is that working?
That's great.
Okay, cool. I can't see anybody now. So anyway, all right. Well, thanks so much for having me, Spencer, and to Meg for the invitation. And it's great to be with everybody and talk a little bit about what my organization does and a little bit about what I've done. I know this is geared towards kind of 8th to 11th grade, and so you want to get a little bit of background on sort of being a scientist. And I won't talk much about sort of my path, but happy to discuss any of that in the Q&A or afterwards. If anybody has any questions, I'm easy to find through email, through my organization. So just real quick about the Tahoe Institute for Natural Science, or TINS, as many know it. We are a research, education, and outreach organization. We have a longer-term goal of bringing a world-class interpretive nature center to the region. And this doesn't exist. This is just me playing with Photoshop. But we do engage in a lot of programming, and we do a lot of supplemental science curricula for local school groups and field trips and in-school programs. We've had over 53,000 student contacts in the last 10 years, and lots of free guided outreach, nature programs, bird walks, wildflower walks, talks, festivals, that sort of thing. But research, and the research that we do, informs all that content. And one of the things that makes us a little unique in the area is how much research we do. I've been really fortunate in my career as a researcher to work with lots of interesting critters and in some pretty interesting systems. I worked in Cameroon in the deepest, darkest jungles of Africa on hornbills and monkeys and fruit tree interactions for a while. I have worked with white sharks off the coast of California and trolled for them with surfboards and stuff like that. Pretty fun. I've rigged up the forest with surveillance cameras to see what kind of predators are eating bird eggs and nestlings. I've done some agility training and experiments with chipmunks. All kinds of fun stuff that has led up to when we started the Tahoe Institute for Natural Science 10 years ago. We conduct a lot of different types of research at TINS, and one of the nice things about being in charge of that is I can kind of follow my curiosity, and whatever I can find funding for and sort of justify, we're able to pursue. And so that's been really, really gratifying. One of the things we started right off the bat was a long-term mist net study where we put up nets and catch birds that are using this area during post-breeding dispersal and early migration. And so we've been doing that at a few sites for 10 years. We also do a lot of citizen science, get the public involved. And so we coordinate Christmas bird count. We coordinate dragonfly weekend, where we go out and just learn about our local dragonflies and damselflies around here. We coordinate a local butterfly count. We've done work in the Angora fire burn related to salvage logging and woodpeckers and bluebird species there. Just natural history stuff, lots of discoveries. This is a Mount Lyell salamander, which we're looking at just local distribution of that species a little bit. Investigating the effects of dog exclosures, beach closures to dogs in South Lake. That was a number of years ago, but pretty interesting. A couple years ago, I did a little research on yellow jackets and some bait experiments there. Most of the work I've done in Tahoe for 20 years has revolved around aspen and bird communities in aspen. And so that's what my dissertation was on, bird communities and aspen stands and how conifer encroachment brings in predators and affects nest success. But lately we've had a moth that comes in and eats all the leaves off the aspen trees. This is a moth from Europe or Eurasia. And anyway, so we're again looking at nest success in the aspen, but now as it relates to defoliation, thanks to these moths. So that's what a lot of the research I'll be doing this summer is on. But I'm going to talk today about just a few different research projects, a little bit more discrete projects that we've worked on, and just show you guys some of that diversity and go into a little more depth on that. And I'll focus on tiger beetles, Swainson's thrush, and montane rabbits. So montane rabbits, leporids, is part of the lagomorphs, really, really important group of animals, and they've been completely ignored by almost all the monitoring efforts that have happened at Tahoe. Tahoe's had a lot of pretty intensive biological monitoring over the last few decades, and nobody looked at the rabbits, which is kind of crazy because there's a lot of them. They're in a great diversity of the habitats around here. They eat a lot of vegetation and sort of shape vegetation structure. And as a decent sized animal that there are a lot of and it's active through the winter, they're super important with regards to predator communities, and especially predator communities making it through the winter. You don't get wolverines and Sierra Nevada red fox and lots of other species without them having something to eat through the winter. Well, rabbits stay active through the winter. Sierra Nevada red fox needs to eat that. So probably very important species for the foxes. Sorry, that's such a cruddy photo, but anyway, we have 4 species in the Tahoe region. And the top one there, Nuttall's cottontail, is an interesting one that we're also looking at right now. It used to be just restricted to the very easternmost part of the Tahoe Basin. And then in the early 2000s, kind of came out of the mountains and spread across both North and South Lake over about a 15-year period. And about that same time, we started to get gray fox for the first time ever in the Tahoe Basin. So that's a cottontail in my yard back when I had a lawn on the left, and then you can see the gray fox there. The first record for the Tahoe Basin ever was 2005, just after the cottontail kind of exploded. So interesting stuff there. And we're trying to get to the bottom of what caused that expansion, or allowed for it anyway. Also, just want to point out we have snowshoe hares here. And then occasionally we'll get black-tailed jackrabbits. I've seen black-tailed jackrabbits right up towards Donner Summit as roadkill along I-80. So they don't spend much time here, but they do pass through some of the more open habitats. So, but cottontail, snowshoe hare, and white-tailed jack, the main three. And lastly, rabbits are famously fecund. They make more rabbits, right? They reproduce a lot. Some of the older students may have seen figures like this showing snowshoe hare and lynx pelt records from the Hudson Bay Company, and sort of showing this 1 to 2 year lag of these population cycles that go up and down. And so anyway, just speaking to how important they are to predators, right? So back in 2010, the winter of 2010-11, I started a project mostly over that winter that was looking at white-tailed jackrabbits. This is a white-tailed jackrabbit. They are very large Alpine jackrabbits. You will note that it is white. They do turn white in the winter. And they tend to live quite high. They like open habitat. They were really common. They were the dominant, believed to be the dominant jackrabbit in the northern half of the Great Basin before cattle grazing and black-tailed jackrabbits sort of took over that changed landscape. But their populations have declined quite a bit and appear to be declining all over the continent where they occur. There were prior to 2010 kind of no documented records for decades, and this gigantic 2-volume report here is the Lake Tahoe Watershed Assessment, and it came out in 2001. It was meant to be sort of a summary of all ecological knowledge that related to Tahoe at the time, and they declared that white-tailed jackrabbits were extirpated from the Tahoe Basin, which is to say they'd gone extinct locally, right? So we didn't have them anymore. And Nevada Department of Wildlife believed that they were gone from western Nevada as well. Well, as a backcountry skier, I knew that wasn't true, that they were around. But people hadn't been looking. They live up high, usually above 8,500 feet. They're shy, and unless you have a dog that kicks them out, you're not likely to see them. They're nocturnal. People see a white rabbit, they usually just assume it's a snowshoe hare, and nobody had really been looking. But people should be looking, because since they live at the tops of mountains and high ridges, there's a lot of potential for these things to really suffer from climate change. We know that they're in great decline as it is. So if their habitats get squeezed right up off the top of the mountains, where do they go, right? So we wanted to do a study to confirm and document that they were still here, try to understand sort of their baseline distribution around Tahoe, see if we could estimate, get some sort of sense of population. Are they really rare, or are there a lot of them, or can we get real estimates of densities, see if they had any predictable habitat associations, maybe certain vegetation types. And the way we're going to do this is go out and look for them, look for their sign, and where we found sign, to put out cameras. Now, I did this 10 years ago, so still actually was using some film cameras back then, as well as trophy cams. I think folks at Headwaters, you guys do some stuff with trophy cameras, digital game cameras, so you may be familiar. This work happened in the winter, and so just a bit about working conditions at a lot of creek crossings, icy slopes, gigantic overhanging cornices, and a lot of wind. And you can see in the bottom right photo just big pile of equipment. I was skiing around with, 30, 40, 50 sometimes pounds worth of camera gear and batteries and stuff, setting up these camera traps, and then I'd try to leave them out for a week or so. So that meant, going straight up chutes and gullies like this at Mount Rose, and it was maybe the most physically demanding field work I think I'd ever done. So why did we do this in the winter? Well, it's a lot easier to find tracks of pellets in the winter. The snow covers up most of the vegetation, so that means there's less cover, which means it's easier to, the animals get concentrated in fewer little patches of cover. A lot of animals hibernate or otherwise, under the snow for the winter, and so you don't waste your film on a film camera, batteries, taking pictures of stuff you're not really as interested in. And it's a lot more efficient to get backcountry, and the snow kind of smooths out the landscape, and so you can ski around, I think, a lot easier. So anyway, we'd go out, and first thing is just try to find pellets and tracks. And thankfully, white-tailed jackrabbits have a very distinctive track. They gallop always, even if they're just sort of walking around. So what happens here, if it's a little more clear on the right-hand side, I don't know if you can see my mouse, but here's a pair of front feet. The animal is heading in both of these, the animal's heading towards the top of the photo. And what you have is they put their two front feet down, and then the hind feet come over what's called an overstep, the long rabbit legs, right? And place their feet down, and then they bound to the next one. Well, with a snowshoe hare, which occurs in a lot of these same habitats, they put their hind feet down at the same time, almost always. And so you get these two hind feet. The big feet are kind of evenly placed, perpendicular to the direction of travel. Whereas with a white-tailed jackrabbit, they're always going to be offset, one in front of the other. So maybe that makes sense. So anyway, pretty easy to find those most of the time. And so then there's jackrabbits around. Pellets can be kind of hard to find. Here's one here on a snowy slope, and here's one up on Quartz, up on Job's sister, South Lake. And they're round, so they do blow around a little bit. So it's not as meaningful if you find a pellet. But if you find a big pile of pellets, then that's likely a spot where they spend a lot of time. Maybe that's a good place to put your camera trap. Now, these pellets are really quite large, and so some of them can be up around 17, 18 millimeters. They do tend to get a little bigger as they degrade with weather, but the smaller pellets on the right of each column are just are cottontail pellets, which are pretty good size. And then you can see how much larger the jackrabbit pellets are. They look like elk pellets almost. Actually, they look a lot like elk pellets. So we were sort of confident that just size alone would be diagnostic, but I mentioned the black-tailed jackrabbit, and we did get some black-tailed jackrabbit photos at some of our sites. And so that they also have very large pellets. And so the only way to know if our pellets came from which species was to do mitochondrial DNA genetic work with a lab out of UC Davis, which we did as sort of a proof of concept thing. And that led to a whole bunch of other stuff, which I won't talk about today. But anyway, more interesting work. But it was disappointing that we couldn't just know that a pellet meant white-tailed jackrabbit because it was big. So here's something you probably didn't know. Rabbit urine is kind of orangey red. It fades to brown and then yellow, but it starts off really looking like blood. So it can be quite alarming when you find that on the snow surface. But anyway, more sign, right? So just a few pictures of some of the setups. So, thinking these rabbits might be using trails, try to face the camera across a trail or up against cover like this, I believe that's juniper. And the rabbits really like some scenic places. This is up on South Camp Peak, looking across at Emerald Bay there, and right on top of Friel Peak, the highest point. There were rabbit pellets just right up top. Pretty cool. Had a lot of complications with my digital cameras. We really had to dial in the exposure. What happens is you've got these LED flashes, and it's a big white rabbit and white snowy background, and it just gets blown out. And you get no detail, right? So I ended up using masking tape to cover up most of the flash, and eventually got some really nice images. But that took some trial and error, and that meant a lot of trips, a lot of skiing back and forth way out there. So it's kind of hard when you put your camera out for a week and then you go back and your images all look like this. You have to try again. So that was also a really big snow year. And with film cameras in particular, well, your cameras aren't capturing anything if they're under the snow, but also you use up all your film. So this was really frustrating. Part of that study was just putting cameras out, and it would snow 2 days later and you'd get nothing. And then you got to find your camera and dig it out. This one, I think I had to dig down 4 feet to find. This is up by Little Round Top. And also the landscape really changes as it snows, and so it can be really confusing and hard to figure out where your camera even is. I think this took me 45 minutes to even find my first stake. This is up on Mount Rose. That's Church's Peak in the background. So this is well up over 10,000 feet. So, a lot of this kind of thing. Rye mice. Here's a camera here, film camera here. Your equipment just does not work when it's covered up by ice like that. Got a lot of fun bycatch. There's a short-tailed weasel up on the top left, bobcat top right. And we got a lot of dogs and a lot of hikers, a lot of curious hikers, but most of our equipment didn't get messed with too much. Okay, results. Nice thing is that we found persistent white-tailed jackrabbit, still there most of the places we expected them, and so that was really encouraging. And in fact, they were very high densities down around Carson Pass and in the southern part of the Tahoe Basin. The big surprise was that we didn't find any at all in the Desolation area, and they were there 50, 60 years ago. So we're trying to kind of still speculating why in the world they might have disappeared from there. But anyway, nice to know that they still persist and need to be taken into consideration for ski area development and telecommunication development and climate change and so forth. Anyway, we sort of found them where we expected them for the most part. We identified some plant associations. They may not like decomposed granite where it's really coarse. And interesting thing we found on in the Carson Range on the east side of the lake, they're actually able to drop down to lower elevations very easily, and so they do so. They kind of disappear for the winter. But here, around the Carson Range, they actually move upslope, where the wind strips the snow off vegetation so they can access it. So that was pretty cool, interesting to see. Still don't know why they might be gone from Desolation, what might have caused that. And I won't bother you with metapopulation dynamics. That's something we're thinking about, though. So here's a picture. Most of them, you just get this big, lanky, leggy rabbit blasting through the frame, but sometimes they park it right down in front of the camera, which is nice. Sometimes they get right in front of the camera. But here's sort of the classic look. It's a big jackrabbit, long ears, longer than the head, and then you can see a white tail sticking out. On a snowshoe hare, you won't see the tail because it's tiny. We do have Nuttall's cottontails at a lot of these higher elevation sites, and so more of a classic bunny shape. And just to give you a look at a comparison of the size of these things, this is a, same background. You see these rocks in the same place, and you can see that the white-tailed jackrabbit is just gigantic in comparison. Cottontail, maybe 2-3 pounds. Snowshoe hare, maybe 5 pounds. Maybe a big black-tailed jackrabbit could be 7-8, and whitetails can be 10 or more pounds. So they're really gigantic. They look like a horse sometimes. And I like this 2-fur shot that I got up on Mount Rose, starting to come out of its winter pelage. If you ever want to see these, they're really shy. If you flush one up, they tend to just run over the nearest horizon. But if you go to Tuolumne Meadows near Tioga Pass, they're actually quite tame because nobody has dogs in the park. And so you can kind of walk right up to them. This is what they look like in the summer. So pretty great. I'm going to skip ahead this slide, but we're continuing to work on those and snowshoe hare and looking at conservation genetics. And that's with UC Davis and Ben Sachs lab at UC Davis. Swains and Suresh. Okay, next thing, shifting gears. This is a bird that used to be maybe never common, but very widespread and certainly a major component of the forest community throughout the Sierra Nevada. And starting about 100 years ago, they may started to decline. Long time, right? And so we wanted to determine the current breeding status and distribution throughout the Sierra Nevada. This has been sort of an ongoing interest of many other researchers as well. Is the population still in decline? And most importantly, what caused these declines? The habitat seems to be in pretty decent, intact shape most places. So there's a lot of evidence that's sort of suggesting that maybe it was something on the wintering grounds. We know this is a bird that occurs not only through the Sierra Nevada, but well up into Alaska and then all across the Boreal, through the Rockies, New England. So it's all over a big chunk of the country. And different populations have different migratory routes, different places where they stop on migration, and different wintering areas. And some of them may even have 2 wintering areas where they move halfway through the season. So how do you find out where an individual bird or birds from an individual population migrate to? Well, you got to give them a little backpack. So you got to catch them, and then you give them the backpack takes the data, and then you got to catch them again, get the backpack off, download the data. So the little green and translucent things are what's called optical geolocators. And all it is a light sensing, a stalk with a little light sensor on it, a computer chip, and a battery. It's mostly battery. And what it does is it measures light intensity about every 5 minutes, or however you have it programmed. But from that, you can set thresholds where you decide what's day and what's night, and then from that you can get day length, and that tells you how far away from the equator you are, and then you know when noon is, and that tells you how far away from Greenwich Mean Time you are. And so that gives you 2 points on the globe, right, any given day. And then the little black guys with the antenna, those are GPS units. And that's almost entirely battery. It tries to, get a fix with satellites and then records where it is on the planet with tremendous precision. But it can only take 8 fixes. It takes a lot of energy to talk to satellites. And so you can program 10 dates, and it'll try to lock in 8 times. Or store eight fixes anyway. We got a few of these back. They didn't work at all. So it's really disappointing, except for one. We had one work. But anyway, so first thing you do is you kind of program these little gizmos, and you hook them up. These are little, these are leg loops, and it sits almost like a fanny pack on top of the tail. And then you tighten it up, this little bead on the very bottom is a jewelry bead. You crimp it, a little bit of super glue, snip off the end of this like stretchy beading. I don't even know what you call it. The beading string, stretchy, and then it's a pretty comfortable fit. You know, it's just like they're wearing a fanny pack. And these are very, very light. Okay, and then to catch them, I bought some purple martin decoys on Amazon and decided I'd paint them up to look like Swaine thrush. Then you go out to Swaine thrush habitat, which is incredibly thick alder and willow. And you put up your decoy, you cut a net lane to put a mist net, almost like a badminton net of sorts that catches the bird next to the decoy. Here's the net folded up here. You can see my loppers and a machete in the background. A lot of trimming of the vegetation there. And then you lie down under or near the decoy, pull out your iPhone, and play the bird's song. And in doing so, you're basically picking a fight with any territorial Swaine thrush that might be there. And so the theory is they'll come in and attack your decoy and get the net, and then you catch them. And look at that, it works. Yeah, I was pretty pleased with my decoys. I actually had birds kind of investigate the decoys before I even got my phone out once, which was a proud moment. So anyway, here's a Swaine thrush with the geolocator attached, and you really can't see any of it, but just the little light stalk kind of pokes up above the feathers. We did this at 3 locations, 2 in the Tahoe Basin and 1 just outside on the other side of Echo Summit, locally. And then we also did this for 2 populations up in Plumas County, and then a population in Marin. And we wanted to see if, the 3 different groups were going to different areas in the wintertime, and if that might explain why the birds here are in such decline. Birds, I should mention that the birds in Marin seem to be doing great, no declines. So here's what we found. And it turns out that our birds are going all the way down to Colombia and Venezuela. The Plumas County birds are mostly going to Central America, and that the Marin birds are going to Western Mexico. And if you look in the these numbers sort of relate to overall forest loss over that time. And the whole point of that is just to show that these birds are in fact subject to very different vulnerabilities, not only due to migration length. The further you have to go, that's greater risk, but also they're going to very different places that are experiencing different degrees of habitat loss. And so, yeah. Next, we're looking again at some genetics. We pick some tail feathers and look at DNA and see if these are different subspecies and how that might play into this as well. If you've got 2 different subspecies coming together in the same spot, that can lead to some complications behaviorally, and they may not know where to migrate to, and they might show up at the wrong time or sing the wrong song, and stuff like that kind of makes it a little fuzzy. So anyway, here's me looking at different subspecies at Cal Academy. Okay, moving on, lastly, quickly to tiger beetles. I got really into tiger beetles a number of years ago. We had a Tahoe Institute alumnus who, Igor Moroldoyev, who's from Russia, and he knew a bit about beetles, quite a bit, and he wanted to come and work with us for 3 weeks in October, back in 2011 or 12, and he wanted to do wildlife work. And October, it can be beautiful or it can snow the whole time. That's a tough one. I was trying to think, what's going to be active and reliable in October. Well, we picked tiger beetles. We've got about 8 species around Tahoe. Here's 4 of them. They're just really, beautiful iridescent beetles. They're sort of the top predators of their size in the terrestrial realm. They just run down ants and other prey and gobble them up with these big, scary mandibles. And they're really charismatic and fun to watch and just interesting and very cool to work with. So Western tiger beetle occurs on all the beaches and most of the rivers and stuff all around Tahoe. It's a really easy one to find. And they have a spring-fall activity period where they're active in the fall up until it starts snowing a lot. And then they hibernate, as adults, and then they pop out in the spring. And anyway, I had a good hunch that they'd be active in October, so we could check them out. And I thought an interesting study would try to relate how Tahoe yellow crest exclosures affect the beetles. Now, Tahoe yellow crest is a very rare native endemic little mustard on the beaches in Tahoe. It only occurs right on shoreline habitats, a little bit up a few creeks, and just on some kind of back dunes and things. But they're almost all down in South Lake. There's a few spots elsewhere on the lake, and it's the only place on the planet where this little mustard plant grows. And since it's so rare and vulnerable to people stomping on it when they go to the beach, a lot of land management agencies that have this species on their property have built fenced off exclosures. And they look like this. So the idea is that it just sort of keeps beach users out of the the prime yellowcrest habitat, and then the yellowcrest is allowed to thrive, and everybody's happy. So what we thought we'd look at is beetle habitat, beetle populations inside and outside of the exclosures, and we wanted to look at their prey base as well, all the other insects. We had a lot of ideas about stuff we were going to do, behavior experiments and things like that, and looking at larval ecology. And I'm going a little long, so I'll skip past that. The larvae are fascinating, though. You see this little, they dig a little hole and they sit with their face basically just right at the surface of the soil. Something goes walking by, they leap out, grab it, and pull it down. They're a lot of fun to play with. They're pretty cool. But we tried rearing them in the lab and doing a bunch of stuff, and none of it worked out. But anyway, first thing we wanted to get a handle on is what time of day are they active, and that was going to inform some of the other work that we were going to do. And so we did daily activity transects, where we went out every half hour and did a count. And at the same time, we would take air temperature and soil temperature with the little infrared laser thermometer, and also look at cloud cover, wind speed, and that kind of stuff, and get a handle on their activity. And so we found that week that basically they were up and active around 10, and then they shut down around 4 p.m. And the triangles at the bottom is activity there. So that let us know that if we were going to put pitfalls out, the best time was 10 to 4. Civilized, right? Banker's hours. So that's what we did. We put out pitfall traps, which are basically just a little cup of some kind. These happen to be urine specimen cups. But the idea is that the lip of the cup sits right on the soil surface, and that whatever's walking along just drops into the cup. And we put out 100 traps at each experiment. We had 50 traps inside and 50 traps outside, and just like a 1 by 1 meter grid, in the way that we could best fit that out onto the beach. And we left them out there for those 10 to 4 every day. 6 hours. Okay, so our basic hypothesis was that there'd be less disturbance within the fenced off area, and that would affect the soil a little bit, but also lead to more and diverse vegetation cover, and that increased vegetation cover would mean we'd have more prey, a greater diversity of prey, and a greater overall biomass of prey for the beetles. And then that would lead to more beetles. Here's some photos of just kind of the texture of the sand inside and outside. So the bottom half of this photo is inside the exclosure. The top part of the other side of the fence, that's just where people have been walking, and you can see how disturbed that is. Now, people do still get in there, and geese are walking around, and we had to get in there and walk around a little bit. So it's not completely, pristine, but it's a big difference. And you can see it here. Top photo, you've got right side and inside exclosure. On the left, you've got outside exclosure. Excuse me. And if you look very carefully here, you may or may not be able to see this, but the right side's outside, and there's even like about an 8-inch drop in the sand from the inside to the outside. So some of these are pretty dramatic differences between inside and outside. Just look at the difference between like the vegetation and then there's none, right? You can see all this grassy stuff. Here's a little bit trickling out, but okay, well, here's Eager in the lab, dissecting things and counting all the different types of bugs that we caught. And here's a nice bar chart to show pretty strongly the difference between treatment, which is inside the exclosure, and then control, which is just the beach outside of the exclosure, and how many tiger beetles we captured. So this is Meeks, Upper Truckee, Taylor, Tallac, Baldwin, Nevada Beach. And you can see, pretty dramatic. Almost 20, none. 9, 1, 8, 2, 7, 0, 2, 1. And unfortunately, we didn't get any at Nevada Beach at all. And it was such a small sample size that impacts your ability to draw inferences statistically. And so unfortunately, this wasn't what you would say statistically significant, but it's awfully close. With a nice, clean, simple paired t-test, it's pretty nice that you can still do experimental designs this way. Not everything has to be like crazy Bayesian statistics. But anyway, you do want your p-value to be under 0.05, and ours was just a hair over. But with everything that we looked at, our hypotheses sort of played out. So how many tiger beetles were there? Well, there were more inside the exclosures. Total arthropod, prey base, biomass, more inside. Number of individuals of prey, inside versus outside, a lot more in. Prey species richness, that's how many species. I think that is the diversity of the prey. There was much greater, and inside the exclosures, there was more vegetation, and there were more species of plants inside. So anyway, it was a simple thing that we could do in just a couple of weeks in October, and it was a good success. And kind of nice to see too that protecting the yellow crest with these fences actually serves as an umbrella to protect other species as well. So nice to see that. I've since gotten really into tiger beetles and looking at other ecological relationships away from the beaches, but high up on the mountaintops. Some reproductive stuff with a couple other species. Just cool photos of pupa there. Actually looking at some diet experiments this summer, and they're believed to be strictly carnivorous, but it turns out some of them eat corn, but some of them won't. So we're trying to figure out, what that's all about. And even just last week, found a whole new genus for the state of Nevada. This is Omus californicus edwardsii, which is a California night-stalking tiger beetle, not known from the state of Nevada, and apparently they're there. Just this is an adult I found last week. So pretty cool stuff. And I think I need to, that's our website if you want to check anything out. And I will stop this. And there's Spencer.
Perfect. Thank you so much, Will. That was awesome. We have a ton of interesting questions to come on in and share with that. I have a bunch of questions myself too. And so we'll try and work through them here. So the first question I have is, what types of other things do you catch in the pitfall traps when you do those? I know you talked about tiger beetles and some of their prey, but what else do you find in there?
Everything. So a lot of ants, and ants are kind of tiger beetle's favorite prey item. But just about any. I mean, we had a few moths and butterflies, even a few caterpillars would wander their way in. A lot of spiders. Anything that's walking around on the ground might find its way into a pitfall trap. Yeah, but mostly insects. So we were out there kind of monitoring. They're not, a lot of times with pitfall trapping, you might leave them out for a week, or even longer. And so then you'd want to put a little cover over there so that lizards and rodents and things like that aren't making their way in. Yeah. We sat out there for the 6 hours each day to monitor them, just to make sure nobody's dog went sniffing around or people stepping into them and stuff. So, but yeah, any terrestrial arthropod can wind up in there. So we didn't get anything like really cool like a scorpion or something like that, but a lot of ants. Yeah, hundreds and hundreds of ants.
Wow, that's awesome. So if any of you have any questions at home for Will, put them in the comments and we'll do our best to get to them. And then the next question that we have was from, let's see, here it is. Kara's and Ricky, who's wondering about how much do those transmitters on the Swanson's thrushes weigh, and kind of how does that compare to the weight of the bird?
Yeah, so all rigged up with the harness, they're about 0.8 grams, and the birds are at their lightest in the breeding season. And so they're maybe 28, 29 grams. But so during migration, though, they put on a lot more weight. Like a lot more weight. So it ends up being a much smaller proportion, relative to their migratory weight. Nevertheless, it would be like, walking to Columbia with a 15-pound fanny pack on or something. Like, I'm sure they feel it, but they've done a fair bit of meta-analyses at this point, trying to look at impacts on survival. And for most species at that size, it's not an issue. It's negligible. Yeah, the GPS units are about 1 to 1.1 grams, a little heavier. And it'd be nice if that technology can get smaller and more reliable. What happened with ours is that the antennas would break off, or maybe the birds were just reaching back and snipping them off. I don't know, but. So we retrieved a few, but only one had data. The rest were just nothing.
Perfect.
It's really heartbreaking because they're expensive and it's a lot of work trying to put them on there.
So the next question we have is, what's the highest elevation that the tiger beetles can live in the Tahoe area? I know you talked about the beaches and then some of the other meadows that you've done, but what's their range if anyone's interested in studying them?
Yeah, that's a great question. I don't think it has as much to do with elevation as it does. Well, okay, so they are insects, and they're the kind of insects that like to be as hot as possible for within their sort of physiological thresholds to be active. They're very, very fast. Some of the fastest insects on the planet are tiger beetles from Australia, which move something like crazy, like a couple hundred body lengths per second, or just I don't know, but you can see how leggy they are, right? And what they do is they run down their prey, and in fact they're so fast that they go blind effectively while they're pursuing prey. So you'll watch them run on a trajectory, stop, adjust, run, stop, because it just goes. It's a blur, and it's believed that they can't process it fast enough to keep up with their legs. Crazy. Okay, so they need to be a little bit warm, but some species don't want to get too warm. So we have Alpine tiger beetle around here, which is Cicindela plutonica, and they like it pretty cool. And you can find them right up on top of like Red Lake Peak and stuff. But it depends more on the soil that's up there. And so the beetles that are up there are kind of faced with an interesting thing. They need the right soil, they need moisture. And, you know, year to year, the snowpack can have like a cornice, just, you know, you get the snow pockets just persist for months. Well, if the beetles are under there, then it's going to take them a long, long time before they get, you know, a couple drought years where they can pop out. So it's a complicated answer. But, you know, they're as high as the highest peaks around here. And around Downer Summit, you can find that photo I showed of the kind of blue one, and there's California night stalking tiger beetles right around Ambrose's house.
Oh, cool.
So, yeah.
So our next question is a little bit more broad scale, and so you do all this research, and kind of where does it go next? How do you take your findings into the scientific and or policy realm?
Well, it depends on the research. So some of it, do tiger beetles eat corn? Like that's just more of a academic, you know, curiosity kind of a question. But we do a lot of work that's really applied. So a lot of the work we do in Aspen revolves around thinning conifers, and you know, we look and see. Or like the dog exclosures or riparian restoration, things like that. The goal is to help inform management decisions the best way to go about whatever treatments or lack of treatments they might want to do from a wildlife perspective. So, for example, working in the Aspen with conifer thinning, you know, we've found that we sort of expected when you go in a little heavy-handed with a bunch of equipment and cut down all the trees, the conifer trees in an aspen stand, does that have a short-term negative impact on the bird community? And what we found locally here is no, it doesn't. They're actually rebounding really fast, and you know, for the sake of maintaining the aspen, go in and cut out as many conifers as aggressively as you can, and the birds are going to be fine with that. They're actually going to rebound a lot faster. So it just depends on the type of research. It's hard to say where some of the research will go and what it'll lead to. So with like long-term bird banding monitoring, you know, trends and patterns that may not reveal themselves for 20 years, well, you need that long-term data set established to start that. Am I fading in and out?
A little bit, but we can still hear you fairly well.
Okay, all right. Sorry about that.
No, it's great now.
So I hope that answers the question. It just kind of depends. We do try to work with local land management agencies like Forest Service a lot, state parks. Like with with the white satin moth work that we're doing. We're trying to determine a whole nother side question we're looking at is, are the caterpillars themselves impacted by, there's a little anecdotal evidence that suggests that maybe they're not as interested in defoliating trees where there's recently been a burn. Okay, so then we need to kind of look at the chemistry of that. What are the trees doing in response to a burn, and can we use that in an applied way as a management tactic to prevent like widespread defoliation from these moths?
Oh,
you know,
cool.
can we just go in and scorch a few trees and then that helps save them? So we're looking at things like that, and that's definitely applied.
That's neat. I have a couple more questions on the beetles. I think the tiger beetles may have been the fan favorite. The first one from Kenzo was, why are they called tiger beetles? We don't see any stripes on them. And so if there's any reason behind the entomology there.
I'm not sure. I think it's a little bit due to their fierce nature.
Okay.
Since they are a really aggressive terrestrial predator and they just sort of run down their prey, I think it's that. If you look up close, I don't know if I could. Well, never mind. But if you look at a photo of one and their mandibles, you just see these huge, scary teeth, and so it's probably mostly from their behavior. They are a little stripy. They have these distinctive pale patterns where there's a lack of pigmentation called maculations, and a lot of how you identify them is by those patterns. And so they're a little stripy, but not all of them. Yeah.
And so the next question is it could be very broad, but someone was wondering, Rich Chambers, kind of why are there so many different species of beetles out there? If you have any insights, you can answer that on whatever scale you deem appropriate.
Well, according to J.B.S. Haldane, the great Scottish evolutionary biologist, it was because God had an inordinate fondness for beetles. That's a tough one. Sorry, Rich. I don't know how to answer that. I mean, why are there so many orchids? Why are there so many wasps? Just certain groups have been very successful, and evolutionary pressures and selection pressures have led to fantastic diversity of certain groups. Beetles are one of them. Really successful taxon. You know, I don't know why there's so many. Yeah.
Cool. And so I think my last question for you today is, we have a ton of students and scientists watching at home who might be wanting to do their own scientific research in their backyard. And I don't know if you have any tips or resources. If someone wanted to do their own pitfall trap, what might be some ways to set that up for success so they could study what's in their backyard?
Make sure you have permission.
Yep.
That's one. So just you gotta be careful if you're going to be working on state or federal land or anything like that. But if you want to do something in your backyard, pitfall trapping in your yard or more broadly.
Either way, whatever you think is accessible for a high school student.
Right. Get a book. There's a lot of resources out there. So if you're interested in any particular technique, I think it shouldn't be too hard to find available instruction and information on sort of the basics, and collecting insects is a pretty basic, time-honored thing, that is a gateway to all kinds of discovery. And if you think that it's all been discovered already, no way. One of the great things about insects is I've heard it said, if you follow any given random species for about 5 days, you're the world's expert on that thing. Now obviously there's exceptions. People know a lot about tiger beetles. People know a lot about butterflies and certain groups. But if you want to just focus on something and observe it for a few days, you may know more than anybody else on the planet. Observe, follow your curiosity, think about, what questions you might have, and then think about how you might be able to answer those. And then reach out. Most experts, most people that are working in the field are passionate about this stuff, and usually more than willing, if they can find the time, if you're patient and persistent, to answer your questions. And I get questions all the time, and I'm always thrilled to help kind of guide or nudge folks along that want to follow their own questions.
Cool. That's awesome. Just to think that there's an incredible diversity of things, or even insects in your backyard that a lot of people might not know about is pretty cool. And you can look up some methods, like you said, on how to study insects and go out and learn something that maybe no one else knows pretty close to home is pretty awesome and empowering.
Real close to home. A lot of times we grow up thinking that cool biology happens in the tropics, or you got to, I don't know, you got to study some flashy thing or do marine biology or whatever it is. But honestly, there's enough insects and plants and just tremendous ecological relationships and stories to be discovered and unfolded right here in Tahoe, right here in the Truckee area. Tons of discovery just waiting for observant people to come along and uncover. So no need to think that you got to wait until college and then you can go work in India or something. No, man, you can get started right here. You could, learn about buckwheats, learn about the butterflies we have here. There's a lot.
Cool. Yeah. Well, that's awesome. And that probably wraps up what we have time for today. But I did just want to thank you, Will, for answering all these awesome questions that we had from the audience and sharing so much about your research. And so we really appreciate that.
Well, thank you, Spencer. Appreciate it. Thanks a lot to Headwaters, and thanks for everybody who's tuned in or watches later. And it's been fun.
Yeah, and I'm sure if any of you scientists at home have questions on how to do your research, you can definitely reach out to me. My email's on our website, and I'm sure you could probably bug Will too if he's not too busy on the TINS website as well, which has a ton of cool resources also. Cool. All right, well, that's all we have for today. Thank you so much, and we'll see you next week.
All right, thanks.